Just like in animals, plants also exhibit different levels of biological organization where many similar cells combine to form tissues. Various types of tissues work together to create organs, and multiple organs function collectively as part of an organ system. To understand how this organization works in plants, we will examine the structure of a leaf, which is an organ itself. Along with the stem and roots, the leaf forms part of a larger organ system whose primary function is to facilitate the transport of substances throughout the plant's body. When we take a cross-section of a leaf, as if slicing it from the top to the bottom and looking inside, we can begin to observe the different layers and how they contribute to its overall function. Before diving into each structural component, we should first consider the main role of the leaf, which is to carry out photosynthesis. Photosynthesis is a process that requires two key ingredients, carbon dioxide and water, in order to produce sugars, primarily glucose. The water that the plant uses in photosynthesis is absorbed from the soil through the roots and transported upward to the leaves through specialized vascular tissue called xylem. On the other hand, carbon dioxide enters the leaf from the surrounding air by diffusing directly into it through numerous small pores called stomata. In fact, each leaf contains thousands of these stomata, and they are scattered across the lower epidermis, which is a layer made up of epidermal tissue. Once carbon dioxide passes through these tiny pores, it moves into the spongy mesophyll layer. This layer contains loosely packed cells with large air gaps between them, allowing gases to diffuse efficiently and easily through the tissue. From the spongy mesophyll, the carbon dioxide reaches the palisade mesophyll layer, which is where the majority of photosynthesis actually takes place. Palisade cells are arranged tightly together and are packed full of chloroplasts, the structures that contain chlorophyll and capture light energy. Just above the palisade layer is another layer of epidermal tissue known as the upper epidermis. The cells in this layer are almost completely transparent, so that sunlight can pass through them and reach the chloroplast below, where it is needed for photosynthesis. After the leaf completes photosynthesis and generates sugar molecules, these sugars need to be transported to other parts of the plant. This job is carried out by another type of vascular tissue called phloem, which are the green tubes seen in leaf cross-sections. The phloem moves sugars in all directions to supply energy to growing and storage regions of the plant. One major problem that leaves face, however, is water loss. While the xylem continuously brings water up from the roots to supply the palisade cells for photosynthesis, some of that water can escape through evaporation, both from the top and bottom surfaces of the leaf. To reduce the amount of water that is lost from the top, the leaf is coated with a waxy cuticle. This cuticle is a thin, waterproof barrier made of lipids that prevents water molecules from escaping. The greater issue for water loss occurs at the bottom of the leaf through the stomata, since water vapor can easily diffuse out of any opening. Even though the plant needs stomata for carbon dioxide intake, their presence creates a risk of dehydration. To balance this, leaves keep their stomata open only for the shortest possible duration, long enough to allow sufficient carbon dioxide to enter while minimizing water loss. This delicate balance is achieved by controlling the opening and closing of each stoma, the singular form of stomata, through specialized cells called guard cells. Each stoma is bordered by 2 guard cells. When the plant has plenty of water and does not need to worry about conserving it, the guard cells become turgid, which means they are swollen with water. This causes the gap between them to widen, allowing more carbon dioxide to enter. On the other hand, when water is scarce, the guard cells lose water through osmosis and become flaccid. This causes the gap to close, limiting carbon dioxide intake, but conserving precious water. Guard cells have another useful feature. They are sensitive to light. During the night, when photosynthesis is not occurring and the plant does not need carbon dioxide, the guard cells close the stomata to prevent unnecessary water loss. Furthermore, most stomata are located on the underside of the leaf rather than on the top. The underside is more shaded, meaning it is cooler, which in turn reduces the amount of water that evaporates. Lastly, we must mention meristem tissue, which functions as the plant's version of stem cells. Meristem cells are found at the growing tips of the shoots and roots and have the unique ability to differentiate into many other types of plant cells. This is essential for the plant's development and growth, as it allows new structures and tissues to form continuously as the plant matures and expands.